Welcome back, guys. Now let's begin with the new chapter, that is nerve physiology. Okay, now in this video we'll be discussing mainly about the basics. Okay, we are going to see what exactly is a nerve, what are the structures which are present inside a nerve, and what exactly is a neuron and the structure of neuron. Okay, so let's confine this video to the basics. Let's stick to the basics in this video. So let's begin with the topic neuron. And nerve. So what exactly is this neuron and what exactly is this nerve? Okay, where they are present. Now you all know there are nerves in our body. Okay, there are nerves in our body. Where exactly, which nerves I'm talking about? The spinal nerves. 31 pairs of spinal nerves are there and 12 pairs of cranial nerves are there. Okay, now let me draw here the cross section of the spinal cord. Okay, here I am showing you this is a cross section of the spinal cord with the dorsal horns as well as the ventral horns. Okay, now you know from here, see what I am showing. Here I am showing you the dorsal root ganglion. These are the dorsal root. Here is the dorsal root. Okay, here is the dorsal root ganglion, and let's continue this. And one more thing, from here I am showing here these are the ventral roots. So both the dorsal roots and ventral roots, they are fusing and forming this one single structure. So what is this? This is the spinal nerve. Okay, from the spinal cord, who is originating? Spinal nerves are originating. Okay, now for better understanding, okay, let me give you more details. Imagine here is your skin. Okay, imagine this is the skin, your skin. Now someone is coming and touching your skin. Someone is giving the stimulus. Now when someone is coming and touching your skin, what happens? First the receptor is going to be activated. Okay, first the receptor is going to be activated. Now from here, this information about the touch. Okay, the touch is something mechanical. Okay, this is something mechanical energy. Now this mechanical energy will be taken by the receptor. This apparatus called as the receptor is going to convert this mechanical energy into electrical impulses, electrical energy. Okay, so one form of energy is taken in and it will be converted into electricity, electrical impulses. So from here, from this receptor, this information about the touch is going where? To your central nervous system. So from here, there is a neuron. Okay, it's coming like this. Here is the cell body of the neuron. Okay, so this neuron, again it is coming where? This information is coming into the spinal cord, and from here, of course, this information later it will go to the thalamus and from there to the cortex. But the point which I want you to know is, look here carefully, look here carefully. Here this green color neuron, what is it? This green color neuron, it is a sensory neuron. It's taking the information into the central nervous system. The information about the touch, this information is going into the central nervous system. So where exactly is this neuron present? This sensory neuron is present in the spinal nerve. Okay, so first structure which I have shown you here, this structure is called as spinal nerve. Okay, so inside this nerve there are neurons present. So if this is a nerve, if I cut this nerve, inside this nerve, what do you have? You have sensory neurons. They are taking the information to the central nervous system. They are afferents. Okay, and in the same way, let me show you. See here, there is one more neuron. This is the cell body of the neuron, which is where it is present. This is present in the ventral horn. This area is called as ventral horn. Okay, this area is called as dorsal horn. Backside dorsal horns are there and anteriorly there are ventral horns are there. So look here, this neuron, it is coming like this, and this neuron, where it is going? It is going to the muscles. Helps in contraction of that skeletal muscle. So this motor neuron is an efferent. It is coming out of the central nervous system and it is causing the contraction of the required muscles, appropriate muscles. So this blue color neuron which I am showing here, this is a motor neuron. Okay, this green color neuron which I have shown here, this is sensory neuron. Okay, so sensory neuron, motor neuron. So finally at the end of the day, the point which I want you to know is in a spinal nerve, okay, in a spinal nerve, okay, what are there? There are sensory neurons as well as the motor neurons. There are afferents as well as efferents. The sensory neurons take the information to the central nervous system and the motor neurons take the information out of the central nervous system, helps in muscle contraction. Okay, there are efferents as well as afferents. Okay, these green color things are afferents, the blue color things are efferents. Efferents means exit out of the central nervous system. Okay, so finally you have the clarity. So what exactly are the nerves? Nerves are nothing but combo of many neurons. If many neurons are coming together, that's going to form a nerve. So inside a nerve there are many neurons present, both sensory neurons as well as motor neurons. Okay, this is the basic thing which I want you to know. This is same is true with the cranial nerves also. Okay, some cranial nerve have only sensory neurons, some cranial nerves have only motor neurons within them, but the idea is inside a nerve there are neurons. Okay, now let's discuss the cross section. Now I am going to make a cut here. Okay, I am going to make a cut. Okay, means a spinal nerve. This is spinal nerve. For example, imagine here is a spinal nerve. I am going to make a cut. Now what will you see inside a spinal nerve? Or just saying nerve inside, what is present? So this is the cross section. Let me write here. Cross section of nerve. Inside the nerves you are having these fascicles. Again these bundles are there. Okay, these bundles are there. So these bundles are called as the fascicles. And one more thing, these nerves, they are covered. Actually these nerves are having a connective tissue sheath around them. Okay, there is a covering. These nerves, they want to have a dress. Okay, so this dress around the nerves, it's called as epineurium. Okay, epineurium. Okay, nerves are covered by epineurium. Inside the nerves, what do we have? Fascicles. These fascicles, they are also having a covering. They are also wearing their own dress. Okay, these fascicles, they are also covered by a connective tissue layer, a connective tissue sheath. This connective tissue covering which I am showing you around the fascicles, they are called as perineurium. Okay, so this blue color structure which I am showing here, this is perineurium. Okay, perineurium. Next, so what exactly are these fascicles made up of? These fascicles, what exactly are they? See, these fascicles, inside these fascicles, what you have, these are the neurons, these are the axons of the neurons, these are the cut portions of the axons of the neurons. Axons are going like this. Okay, single neuron is going to have an axon. Okay, so what are these things? These are multiple neurons present inside one fascicle. Inside one fascicle there are multiple axons of individual neurons present. Okay, so this is the basic architecture. And one more important point which I want you to know is, see, even these individual neurons, okay, even they are also covered. Okay, they are also having their own dress. So what is this covering called as? See, here I have said that even these individual neurons, they are also having their own covering around them. So this covering which I am showing you here, so this covering is called as endoneurium. Okay, so finally I can say something like this. Nerves are surrounded by epineurium, the fascicles are surrounded by perineurium, and the axons are surrounded by endoneurium. Okay, so this is the basic architecture, and here in between the fascicles, what do you have? There are blood vessels present. These blood vessels are going to give the blood supply. Okay, it's going to give the blood supply to the fascicles, and from there to the axons. Now, as a summary, let me write something like this. Inside a nerve, what is there? Fascicle. Okay, fascicles are there. Inside the fascicles, what are there? Neurons. Okay, neurons, or the axons of the neurons are there. Axons of the neurons are going. The nerves are covered by. Now, look here, the nerves, they are covered by. This is the covering around the nerve. So, this connective tissue sheath is called as epineurium. Okay. Next, the fascicles are covered by perineurium. Okay, and last, the neurons. The neurons are covered by this neurons. Okay, each and individual neuron, they are covered by a connective tissue sheath called as endoneurium. Okay. Now, the point which I want you to know is, around the brain there is a protective layer. Okay, which is called as a brain bridge, like a barrier. Okay, so the brain bridge barrier is a protective lining around your brain, which doesn't allow any toxic substances into your central nervous system. In the same way, even around the neurons, you are having a protective lining, okay, so which doesn't allow any toxic substances or like abnormal substances to affect the nerve conduction. So, what is this called as? This is called as a blood neuron barrier. Okay, just like a blood brain barrier. Okay, I said previously brain bridge barrier, sorry. That's a brain bridge reflex, I just got confused. Just like a blood brain barrier, there is also blood neuron barrier. Okay, so this perineurium, okay, this connective tissue layer, the perineurium, this perineurium is the one which forms that blood neuron barrier. Okay, so the point which I want you to know is, which layer is involved in the formation of blood neuron barrier? It is perineurium. Okay, for example, let me give you an understanding what exactly is this blood neuron barrier. Here is a fascicle present. Okay, here is a fascicle. Okay, here is a fascicle. Now fascicles, they are surrounded by what? The fascicles are surrounded by perineurium, right? Here is these are the perineurial cells. Okay, these are the cells which are forming the perineurium. Now look carefully here, in between these cells, the cells which are forming the perineurium. Okay, in between these cells, there are tight junctions present. There are tight junctions present in between these cells. So because of the presence of these tight junctions, they don't allow any big substance or any toxic substance to pass and enter into the fascicle, because inside the fascicles we are having the neurons, the most important things. Okay, so what the point which I want you to know is, this perineurium, okay, it have what? The cells of the perineurium, they have gap junctions. But because of the presence of these gap junctions, it forms the blood neuron barrier. Okay, and not only that, not only just this thing, here outside the fascicle, what are present? Outside the fascicle, look here, outside the fascicle, this is one fascicle. Outside the fascicle, you are also having the blood vessels. You are also having the blood vessels. Now, for example, let me show you one more blood vessel. Before that, see, here is a perineurium. Okay, here are the perineurium. Now here I am showing you one blood vessel. Okay, so imagine this is one blood vessel. Okay, one capillary you can say. Now even here these blood vessels which are lined by the endothelial cells. Okay, the blood vessels which are lined by the endothelial cells. Now in between these endothelial cells, the gaps between these endothelial cells are also going to be very small, very small. Okay, very less. And this capillary endothelium is also having this tight junctions between them. So tight junctions. So because of this presence of tight junctions, only very small molecules are going to cross the capillary, and only very small molecules are also going to cross the perineurium and can enter into the fascicle. Okay, so the point which I want you to know is this blood neuron barrier is formed by which connective tissue layer? Perineurium along with the gap junctions. And not only that, this blood neuron barrier is also formed by the capillary endothelial cells. Okay, capillary endothelial cells along with the gap junctions. Okay, so these are the MCQs which I want you to know. The MCQ point here is who forms the blood neuron barrier? It's the perineurium. Okay, having said that, now let's discuss about the structure of a neuron. Structure of a single neuron. Okay, structure of neuron. Now let me tell you some basic before going to the neurons. How many neurons are there in our central nervous system? What do you think? How many neurons are there in the central nervous system as well as the peripheral nervous system? There are almost 100 billion neurons are there. Okay, you are having 100 billion neurons. You are a billionaire as far as the neurons are concerned. You are having billions and billions of neurons. Okay, of course you are having trillions of cells in your body, but 100 billion neurons are there. Okay, now if I show you one neuron, you all know that there is a cell body. Here I am showing you typical multipolar neuron. Here is a cell body present. Okay, inside the cell body what is there? There is a nucleus. There is a nucleus and nucleolus. Okay, that's a nucleus. Now to the cell body there are this branching like structures present. What are they? So these are called as dendrites. So to the cell body what are attached in a multipolar neuron? See, there are dendrites present. These dendrites, they acts like an antenna. Okay, antenna is going to grab the signal, right? It's going to receive the signal. In the same way, this dendrites, they are afferents. Let me write here. These extensions are called as dendrites. Okay, they are going to receive the signal from the other neuron. Okay, so they are going to form the synapses. They are going to form the synapses with the other neuron. Okay, dendrites, they are afferents. Means they receive the input, they receive the signal. Okay, now what is this area called as? This area is called as cell body or soma. It's also called as what? Soma. And it is also having one more name called as perikaryon. Karyon means nucleus, you know, peri means surrounding the nucleus, which is nothing but the cell body or the soma. Okay, cell body or soma or perikaryon. And there is this area of the cell body. Okay, there is this area of the cell body. It's still the area of the cell body where there is thickening of the cell membrane. Okay, where there is thickening of the cell membrane. So this area is called as axon hillock. Okay, axon hillock. The point which I want you to know is axon hillock is not a part of axon. It's not the part of the axon. It's a part of cell body. Okay, axon hillock is a part of cell body. Now this axon hillock, it continues as, now it continues as this axon. Here is the axon present, this is the axon. Okay. Now what do we have here? Now here we are having this nerve terminal. These are called as nerve terminal, and these things you can also call as a telodendria. Okay, telodendria. Okay, telo means last, final. Dendria means like a dendrites. Okay, telodendria. From here, the neurotransmitters will be released. Okay, now this telodendria, what they forms? They forms the synapse. Okay, they forms the synapse with the dendrites of other neurons. They form the synapse with the, synapse means connection. They forms the synapse with the dendrites of other neurons. These are the telodendria or axon knobs. You can also say axon knob. Next point which I want you to know is the first part of the axon. Okay, the first part of the axon. Okay, this 50 to 100 micrometer area. The first part of the axon is called as initial segment. Why these names are important? Because there are MCQs related to them. Okay, the first part of the axon is called as initial segment, and in this axon, by this time in this axon the electrical impulse is being conducted in the form of action potentials. Okay, the action potentials are being conducted in the length of the axon, it. So to provide the insulation, okay, to provide the insulation, the electricity is traveling in the copper wires, so you are having the insulation. Okay, the plastic thing is the insulation. The same way here there is an insulation. So this insulation around the axon, this insulated sheath is called as myelin sheath. Okay, this is called as myelin sheath. Next, in between this myelin sheaths, there are these gaps present. Okay, there are these gaps present. These gaps, okay, small gap, 1 micrometer gap. Okay, this gap is called as nodes of Ranvier. Okay, so the nodes of Ranvier. So these are some important points which I want you to know. Just the basic terminology which I want you to know. Basic structure. If you go into details within the cell body, in the cell body, what are there? There is a nucleus. Okay, there is a nucleus, and there are also structures present here. Okay, there are these structures, aggregated structures, which are present here inside the cell body. These structures are called as Nissels granule. Okay, Nissels granules. What are these Nissels granules and what are they made up of exactly? See, these Nissels granules, they are aggregates of, these Nissels granules are nothing but they are aggregates of what? Rough endoplasmic reticulum. Okay, they are aggregates of rough endoplasmic reticulum along with its ribosomes. Okay, along with what? Ribosomes. Rough endoplasmic reticulum means it's the endoplasmic reticulum along with the ribosomes which is involved in the production of the proteins, it. Okay, so in the neuron cell body, this aggregation of the endoplasmic reticulum is called as the Nissels granules. What are some important points which you should know for your exams is, these Nissels granules, they are only present in cell body. They are only present in cell body or soma. They are not present, they are never present in the axon. Okay. Now, when a neuron is dying because of a stroke or some other reason, ischemia, when the neuron is dying, these cells granules, they will also undergo dissolution, they will separate. Okay, so this process is called as chromatolysis. Okay, let me write here. What exactly is chromatolysis? Chromatolysis means it's a dissolution. Dissolution of cell bodies during injury. During nerve or neuron injury. Okay, so during injury time there will be chromatolysis. That's the histological finding which you can have. Okay, what else you should know for your exams is the cell bodies, size of the cell bodies. Usually the cell body size, if you take an average, okay, let me put it this way. The cell body size is the smallest in which neuron have the smallest cell body and which neurons have the largest cell body. So let me write here itself, smallest cell body. Okay, smallest in granular cells. Where are these granular cells present? Granular cells are nothing but the neurons. Granular cells are the neurons which are present in cerebellum. Okay, so cerebellum have smallest cell bodies. Okay, cerebellum have the smallest cell bodies which are granular cells. Next, largest cell bodies are present in the cell bodies of motor neurons. In spinal cord. Okay, so you know, in the ventral horn of the spinal cord, just now I have shown you, for example, look here. See, this is the ventral horn of the spinal cord. I have just explained this is the ventral horns of the spinal cord. Here who originates? It's a motor neurons which are getting originated, the motor neurons. So these cell bodies, they are largest in the human body. Okay, they are the biggest. Okay, they are the biggest. So smallest are the granular cells of the cerebellum and largest are the motor neurons of the spinal cord. So now what else you should know regarding the basic structure of the neuron is, I have shown here the myelin sheath. Okay, myelin sheath, the myelination, it's giving the insulation, right? It's giving insulation. Now this myelin sheath, it is formed by which cells? Okay, neuron is having this myelination. Who forms this myelin sheath? Myelination for the neurons in the central nervous system. If a neuron is present in the central nervous system, okay, if a neuron is present in the central nervous system, that is the brain and spinal cord, myelination is done by cells called as oligodendrocytes. If a neuron, if it is present in the peripheral nervous system, that myelination is done by a total different cell, that is called as Schwann cell. Okay, so myelination is done by oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. Is there any exception for this? There is one exception, that is optic nerve. Exception is second cranial nerve, that is optic nerve. Now here itself it is a nerve, optic nerve, it's a cranial nerve. Cranial nerve means peripheral nervous system, so it should be lined by Schwann cells, but no. Though it's coming under cranial nerve, it is lined by, the optic nerves are lined by oligodendrocytes. Okay, so that's why in a condition called as multiple sclerosis, okay, in multiple sclerosis what happens? In multiple sclerosis, the myelination will be affected. Okay, the myelination, the antibodies are produced against the oligodendrocytes. The myelination in the central nervous system will be affected. But in multiple sclerosis, the patient is going to have visual problems. You might think why? So just now you told multiple sclerosis is a problem of central nervous system. The myelination in the central nervous system will be affected. Okay, the antibodies are produced against the oligodendrocytes. I have told you, optic nerve, though it is a cranial nerve, okay, though it is a cranial nerve, it is lined by oligodendrocytes. That's why in an autoimmune disorder called as multiple sclerosis, second cranial nerve, optic nerve will be affected. So the patient is going to have ophthalmoplegia and visual disturbances. Okay, the patient is going to have visual problems, visual related problems are going to be there. Okay, so basic things completed. Now, anything else? See, I have told you nodes of Ranvier. Okay, nodes of Ranvier. See, nodes of Ranvier are the gaps, they are the small gaps between the myelin sheaths, right? In this nodes of Ranvier, okay, I haven't discussed with you what exactly is the action potential, but later I will tell you. The action potentials, this electrical impulses are getting jumping. Okay, they are jumping from one place to other place. This is called as a saltatory conduction. The electricity is jumping. Okay, this is called as a saltatory conduction of the action potential, which we will discuss in the next video. Okay, what is resting membrane potential? Later we will discuss about what is an action potential, saltatory conduction. That will be discussed in the next videos. How this electricity is transmitted in this axon. Okay, but for now you should know, in the roads of Ranvier, this electricity is jumping. For that process, what do you require? You require here more channels. Which channels are present here in these nodes of Ranvier for the saltatory conduction? You require voltage gated sodium channels. So in the nodes of Ranvier, there are maximum number of voltage gated sodium channels. So maximum number of voltage gated sodium channels are present. Okay, so in the nodes of Ranvier, the electrical impulse is being transmitted. Okay, the conduction is happening. Now, have you ever heard about drugs called as a lidocaine, xylocaine things? What these drugs will do? Lidocaine, for example, I am talking about lidocaine or lignocaine. Lidocaine. What exactly is this? It's a local anesthetic. Okay, it's a local anesthetic. You can use it in the other forms of anesthesia also. Now this lidocaine, what it will do? It blocks voltage gated sodium channels. Okay, in the nodes of Ranvier. So what happens if the voltage gated sodium channels are blocked? I have told you, these voltage gated sodium channels are helping in the saltatory conduction. Okay, in the saltatory conduction. Now do you think the nerve impulse conduction is going to happen? Now do you think the electrical impulse is getting transmitted in the axons if the voltage gated sodium channels are blocked because of a drug called as lidocaine? No. So whatever you do, the impulse conduction is not happening. Okay, the impulse conduction, the information is not transmitted to the central nervous system because these voltage gated sodium channels are blocked. Now impulse conduction is inhibited. So that's why you cannot feel anything. Okay, you have inhibited the transmission of electrical impulse within the axon by blocking the voltage gated sodium channels. So you are having the anesthetic effect. You can't feel anything. Whatever you do, you can't feel it because that information is not passing. Okay, so that's the one more point which I want you to know. Next, where exactly is this action potential? Okay, action potential is generated. Okay, action potential, the fluctuation of the membrane voltage. Okay, the conduction of the impulse. Action potential, where it is initiated. See, the answer for this is there are two types of neurons, you know. One is sensory neuron and motor neuron. Action potential generation. For a motor neuron, Okay, for a motor neuron, action potential is generated in the initial segment. Okay, in the initial segment, action potential is generated. For a sensory neuron, action potential is generated in the nodes of Ranvier. Okay, so these are also MCQs which I want you to know for your exam. Okay, you might be thinking why action potential generation is different for a motor neuron and sensory neuron. For example, look here in the spinal cord. Okay, here in the spinal cord. Now, here the blue color neuron which I am showing, here is the motor neuron, you know it. Now, what exactly is this area which I am pointing? Okay, let me draw a new diagram so that it will be better for you. Here is the cross section of the spinal cord. Okay, now from here which neuron is starting? This is a motor neuron that is coming out. Where this motor neuron will go? It will go for the neuromuscular junction, for the contract of the muscle. Now, what is this? First part of the axon. The first part of the axon is called as the initial segment. Okay, from here, what are these things? These are myelin sheaths. The space between the myelin sheaths are called as nodes of Ranvier. Okay, now in this area, okay, in the first part, in this area, okay, the action potential will be generated, and that action potential is going to be jumping like this into the nodes of Ranvier, and it is being transmitted. Okay, but look at the sensory neuron. Here is your skin. Okay, here is your skin. Now, when a stimulus is coming, that stimulus will be detected by the receptor. Now, who is attached to the receptor? Sensory neuron is attached. Here is a sensory neuron. Okay, so sensory neurons are projected in this direction. Sensory neuron. So, this sensory neuron, here is the cell body of the sensory neuron. This is one cell body. This sensory neuron is coming like this into the central nervous system. Okay, now what are these things? Now I am showing here, these are the myelin sheaths. Now you tell me where the action potential should be initiated. The action potential should be initiated at this area, because this is near to the stimulatory point. Here you are stimulating, okay, there you stimulated. So immediately here the action potential should be generated, and it should be jumping here, and it should be jumping here, and later it should be jumping here. From here it should be going into the central nervous system, just like that. So what is this? So here is the area of, here you are having maximum number of voltage gated sodium channels, that's the nodes of Ranvier. So in a sensory neuron, it's the nodes of Ranvier where the action potential is generated, not the initial segment, okay, not the initial segment. Okay, so that's the one point which I want you to know. Now, after this, what else? Types of neurons. The next thing is types of neurons. Based on the function, you know, there are sensory neurons, there are motor neurons, but I am talking about the anatomy, okay, histology. There are three types of neurons. Actually, there are four types of neurons. Three main things. What are they? See, there are four types total, four types of neurons based on the histology. First type of neuron is called as unipolar neuron. Okay, unipolar neurons, which are usually not present in the humans. According to physiology, they are not present in the humans. But one question is there: unipolar neurons are present in which area in a human? Okay, according to the anatomy. So unipolar neurons means what? How the unipolar neuron is going to look like? Unipolar neuron means, see, here is the cell body. There are no dendrites. There is only one process present. Okay, it's having only one process, one axon. That's it. So these are called as the unipolar neurons. Where exactly are these unipolar neurons present? They are present in the mesencephalic nucleus of Fifth cranial nerve, that's the trigeminal nerve. Okay, so the mesencephalic nucleus, actually the fifth cranial nerve, it's having the largest nucleus. Okay, there is a central pontine nucleus, there is a spinal nucleus, and there is a mesencephalic nucleus. There are three nucleus. Actually, there are three nucleus present for a trigeminal nerve. The mesencephalic nucleus inside the mesencephalic nucleus, there are a unipolar neurons present. Okay, unipolar neuron means it's a cell body with only one axon. That's it. Next one is a pseudo-unipolar neuron. So where are the pseudo-unipolar neurons present? First of all, what does it mean by pseudo? Pseudo means false. Okay, why it is called as false? Because see, here is a cell body, here is a cell body, and there is only one process coming out. Okay, so you are thinking like it's a unipolar neuron, but this process is going to divide into two processes. Okay, where you have seen, just look here, this is a sensory neuron. Here I am showing you this is sensory neuron which is taking the information into the spinal cord. So these are sensory neurons, okay, which are present here also. Let me show you. See, in the beginning also I have told you. So there is cell body with one process, and one process towards the central nervous system and the other process is towards the receptor. So these neurons which are present in this area called as a dorsal root ganglion. Okay, these sensory neurons which are present in the dorsal root ganglion, they are examples of pseudo-unipolar neurons. So where are they present? The sensory neurons in dorsal root ganglia. Okay, they are pseudo-unipolar neurons. Then next type of neuron which I want you to know is a bipolar neuron. In bipolar neurons, look here, here is a cell body, there is one extension going in this direction, and two processes, means directly from the cell body, two processes are coming. Okay, here are dendrites. So such neurons are called as bipolar neurons. Where are these bipolar neurons present? These bipolar neurons are present in this FMGE question and NEET PG question. They are present in retina. You know, in the visual pathway. Okay, there are 10 layers in the retina. There are 5 different types of neurons in the retina. In the visual pathway, first order neuron, second order neuron, third order neuron. First order neurons are rods and cones. Second order neurons are bipolar cells. Bipolar cells acts like a second order neurons. Okay, so the point which I want you to know is bipolar cells, where they are present in retina. Okay, not only there, in olfactory epithelium. Okay, in olfactory epithelium, bipolar neurons are present. Next, last one is multipolar neuron. Okay, multipolar neuron. Where are these multipolar neurons present, sir? Multipolar neurons are many. There are many multipolar neurons. Most of the neurons in your body are multipolar neurons. For example, the motor neurons. Okay, the motor neurons from the ventral horn of the spinal cord. They are the multipolar neurons. They are having, like, you know, what exactly a multipolar neuron is? This neuron which I have shown you here. This is a multipolar neuron with one axon and multiple dendrites. With one axon and multiple dendrites. If our cell body is having multiple dendrites, it is called as a multipolar neuron. Okay. Now after this, what I should teach you? Axonal transport. The next topic which I want you to know is axonal transport. Okay. So what is the concept of this axonal transport? Now let me draw a cell body here. One more neuron. Here is a direction. For example, here is the cell body and this is the axon. Here is axon. Here is a nucleus. These are the dendrites. Okay. Now I am asking you, Where are nissl granules present? Nissl bodies, where they are present? These nissl granules are present in the cell body. Okay, what they are making? They are making a protein, or they are making a neurotransmitter. Okay, now that neurotransmitter, where it should go? That neurotransmitter which is getting produced, okay, which is getting produced in the cell body, it should go into the nerve terminal. It should come into the nerve terminal, right? So that the neurotransmitter will be released. So how a substance which was produced here, for example, here a protein is produced, like here a protein is produced, a neurotransmitter is produced. How that protein will be transported in this axon, how it is going to be transported in the axon, and how it is going to reach into the nerve terminal or the axon knobs? How? Means there are highways present. Highways are not only present in the country, these highways are also present in the axon, okay, in the cell also. So there is this highway, okay, which is made up of a protein called as a tubulin. Okay, roads or highways are made up of cement, tar. In the same way, this highway is made up of tubulin. This tubulin is forming. What is the name of this highway? This is called as a microtubule. So this green color thing is microtubule. What exactly is this microtubule? It's a cytoskeleton. Okay, it's a cytoskeleton. Microtubules, microfilaments, intermediate filaments, they're all part of cytoskeleton. So here is this microtubule present. This microtubule, it keeps on growing. Okay, it keeps on growing. It keeps on growing. Okay, as it is keep on growing, it have two ends. One end which will grow along with the axon, along with the axon terminals. So that end is called as positive terminal. Okay, that end is called as positive terminal. And that end which is towards the cell body, is called as a negative terminal. So there are positive end and there is a negative end. Okay, so now on this microtubule, there is a fellow. This fellow is having two legs. Okay, he is having two legs. Now what this fellow is doing? So this fellow is called as a molecular motor. He is a molecular motor. What is he doing? See, this molecular motor, in the name itself, it's a molecule. It's helping the motor movement thing. So the protein which is getting produced in the cell body, that will be taken by this molecular motor. Okay, the vesicle will be taken by this molecular motor, and that vesicle will be transported towards the positive end. It's going to move. This molecular motor is going to move towards the positive end. Okay, so this type of movement is called as, this movement towards the positive terminal, this is called as anterograde transport. It's called as what? Anterograde transport or anterograde cargo. Okay, you can say. And there is one more fellow. Okay, what he is doing? He is moving the substances. Okay, he is moving the substances in the opposite direction. He is moving. Okay, on a highway, there are two. Okay, in the middle there is a divider. Okay, in the middle there is a divider. There are cars moving in this direction. There are also cars moving in the opposite direction. Okay, two-way transport, right? The same way, on this microtubule, there are two fellows present. One fellow is moving towards the positive end. Other fellow is moving the substances towards the negative end. Okay. So the name of this red color fellow, okay, the molecule which is helping in the anterograde transport, this red color fellow is called as a kinesin. Okay, and this pink guy who is moving in the opposite direction, helping in which kind of transport? Retrograde transport. This guy is called as a dynein. Okay, dynein. So dynein and kinesin, these two are molecular motors. Okay, they are molecular motors, dynein and a kinesin. Now, the MCQs which you should know for your exam is, with how much speed this kinesin is moving, with how much speed the dynein is moving, means retrograde cargo or the anterograde cargo. What is the speed? The speed of this anterograde transport. Okay, let me write here. Anterograde transport, the speed I want you to know. It is 400 millimeters per day. Okay. And what is the speed of this retrograde transport, the fast retrograde transport? With how much speed, with how much fastness it's going to move? It's going to be 200 millimeters per day. Okay, 400 millimeters per day and 200 millimeters per day. Okay, this is the question which was asked in the exam. This is the question. This is the question, 200 millimeters per day. So, integration. Now, there are certain toxins. Okay, there are certain toxins or viruses. Okay, there are toxins and viruses which enters into this dynin. Okay, they are going to come and bind with this dynin. For example, imagine there is tetanus toxin, or like for example, a rabid dog comes and bites here. Okay, rabid dog comes and binds here. Okay, now when a rabid dog comes and binds here, the rabies virus is going to enter here first. Okay, now this rabies virus, the rabies virus will go into the central nervous system, hippocampus, you know the thing. Now when the rabies virus, how it's going to reach your central nervous system, via the blood? No, the rabies virus first, it will enter into the neurons. Now after entering into the neurons, this rabies virus goes and binds with the dynin. So this dynin molecule, it's going to help in the retrograde transport. Okay, now the neurons are present from here till to your central nervous system. So with the help of this reverse axonal transport or retrograde transport, certain viruses and toxins will get access into your central nervous system. They will get access. Okay, so that's the point which I want you to know. Next thing is, okay, now a vesicle, for example, a neurotransmitter which was produced here, a vesicle, that vesicle will be transported. See, that vesicle is getting transported via anterograde transport. Okay, via anterograde transport, that vesicle will be transported, and now that vesicle came here. The kinesin is going to release a vesicle. Okay, neurotransmitter here. Now from here to the axon terminal, who will take? See, here are cytoskeletal filaments. Here are the actin molecules present. Okay, here are the actin molecules present. Now from here, that neurotransmitter, this neurotransmitter from here will be taken care by other molecule, other guy. Okay, his name is called as a myosin. So this myosin, okay, this myosin is going to take this neurotransmitter till to the axon terminal. Okay, axon now. Okay, so one more molecular motor is there. He is going to move in both the direction. He moves in both the direction. Okay, bidirectional. So this fellow is called as a myosin. Helps in bidirectional transport. Okay, bidirectional transport. Okay, he is helping in what? Bidirectional transport. Myosin. So finally I can say something. Who are the molecular motors? There are 3 molecular motors present. Who are the 3 molecular motors? This is a question which was asked in the recent exam. Molecular motors are kinesin, dynin and myosin. Okay, kinesin helps in anterograde cargo, anterograde transport. Dynin helps in retrograde cargo, reverse axonal transport, and myosin is bidirectional cargo. Means both the ways. How to remember one thing is the dynin. Recent question, dynin. We are all doctors. Okay, we are all doctors. So dynin helps in retrograde cargo, DR. Dynin for retrograde cargo. Okay. What else you should know is neuronal stem cells. In the basics, neuronal stem cells. Now we know nerves usually won't divide. Okay, they are arrested. Okay, in the Z0 phase they are arrested. Okay, neurons, they won't divide. But there are certain areas in your brain where there are stem cells present. If you stimulate them, they are going to differentiate. Okay, they are going to maintain their own population. They are going to differentiate into neurons. Okay, different types of cells. Now, neuronal stem cells are present in which region? The hippocampus. In the area called as hippocampus, where the memory formation will occur. In the hippocampus, Okay, in this area the neuron stem cells are present. Okay, hippocampus, dentate gyrus, neuronal stem cells are present. Not only here, olfactory bulb and subventricular region. Okay, in this area, subventricular region, olfactory bulb and hippocampus, these are the areas where you can find the neuronal stem cells, and this is a question. Okay, next, pigments inside the neurons. Pigments inside the neurons. Before going to the pigments, I am asking you, neurons, are they going to divide? No, they won't divide. Okay, neurons are not going to divide. They are not going to divide. Who actually helps in the cell division process? See, for example, if I show you a cell which is undergoing cell division, you know, here there is this organelle like this. Okay, here in the center, when a cell is dividing, okay, prophase, metaphase, you know, here a cell is dividing, you know, what forms? Spindle apparatus is going to form. Okay, spindle apparatus is going to form. The spindle apparatus, which is made up of the microtubule, helps in the separation of the chromosomes to the both the extremities. Okay, the spindle apparatus, it is formed by an organelle called as centrioles. Okay, these two things, these are the centrioles. Together they form the centrosome. Okay, together they form the centrosome. But centrioles. Now these centrioles, what they are doing? They helps in the cell division process. They helps in the separation of, they help in the mitotic spindle formation. They help in the movement, okay, movement of the chromosomes. Okay, separation of the chromosomes into two daughter cells. Now these centrioles helps in cell division. Okay, right? They help in cell division. My question to you, are they present in neurons? Centrioles are helping in cell division, but are they present in neurons? Yes, they are present in neurons. Centrioles present in neurons. Okay, they are present in neurons. They help in the formation of the microtubules, help in maintenance of the microtubules, but not in the cell division. They are not involved in the cell division in the neurons, but they are present. Okay, so the next thing is the pigment inside the neurons. Pigments inside neurons. Okay, so in your central nervous system, there are certain areas where they are having some colors. Okay, what are these areas? For example, if I am talking about the substantia nigra of the midbrain, substantia nigra, where it is present, sir? It is present in the midbrain. The name itself is there. It's a substance which is black in color. So why these neurons are black in color? Why these neurons, or why this area which is made up of the neurons? Okay, of course the areas are made up of the neurons. Why this area is black in color? Because of a pigment called as melanin. Okay, because of the presence of melanin. Okay, it is black in color. Next, there is a nucleus called as the red nucleus. Here you are having high amounts of iron. Okay, and there is hippocampus. Here you are having a lot of amount of zinc. Okay, so these substances are present in these areas. Okay, so these are the important points which I want you to know regarding the basics of the nerve and neuron. In the next video, I will be mainly concentrating on the resting membrane potential of a neuron. Okay, resting membrane potential of a normal cell and neuron. Later, if you understand the resting membrane potential, next we can discuss about the action potential and the conduction of electrical impulse in the neuron. Hope the video is helpful. Thank you.